Hello children. So today we are starting another chapter which is tissues. And in this chapter we will learn about the function of tissues, types of tissue, and are they different in plants and animals? We will take up all those topics under this chapter. So what is tissue? Let us first familiarize ourselves. So, tissue is a group of cells that are similar in structure or such cells which come together to work and achieve a particular function. So, one thing is obvious. If it is a group of cells, then tissue will be present in multicellular organisms only. And children, if you remember, prokaryotic cells were those kind of cells which developed first in evolution. And after some time, eukaryotic cells came into existence. And eukaryotic cells had their division of labor due to various cell organelles, and they were more efficient. And this increased efficiency gave rise to grouping of cells, which further increased the efficiency of the entire organism. And that is how multicellular organisms came into existence through evolution. But then, there used to be such simple organisms which had a similar kind of cells forming their entire body. But as the body complexity increased in higher multicellular organisms, similar cells started forming groups which were dedicated for a particular function, and such group of cells forms a tissue. So the concept of tissue is something that it is made up of a group of cells which are modified in such a way that they will work together and do a particular function with greater efficiency. And this is often asked that what is the significance of tissue? So of course, tissue increases the overall efficiency of organisms. So how many types of tissue are we going to learn about? So tissue, for the ease of learning, can be categorized into plant tissue and animal tissue. And these two types of tissue are very different both in structure and function, because we know plants and animals lead entirely different lifestyles. For example, plants are static, they keep standing at one place throughout their lives, while animals have to roam around in search of food, shelter, mate, or to save themselves from predators. So they roam around, and that's why they will have different kind of tissue, and they will spend a lot of energy in this process. But if the plant has to maintain its posture, no matter how high the wind velocity is, if it is raining or it is hailstorms, they need to keep standing, so they need a lot of supportive tissue in their bodies. Another major difference is observed in the mode of nutrition. So plants are autotrophic. They do photosynthesis and make their own food. So there are no different kind of systems required to run around and to search for food and to put that food inside your body and then another system to digest it. No such thing is required over here. But animals are heterotrophic, so they will have a different kind of body structure and different tissue in those body structures just to feed themselves. And then the growth pattern. So the growth pattern of plants and animals are very different. Plants show localized and indefinite growth. So there are certain regions in a plant through which the plant will show its growth, but it will be indefinite. You cannot say that this plant has now stopped growing. In the next spring season it will grow again, and we cannot pinpoint that what will be the duration till which point the plant will grow and after which it will not. While animals, they show uniform but proportionate growth. So all the body organs of animals grow in a definite proportion and uniformly, but their growth is definite also. So after the puberty, the overall bodily growth of animal stops. So animals will not be growing indefinitely, as in case of plants. And now we have understood that plants and animals lead very different lives. They have different growth patterns, different modes of nutrition. So obviously the underlying tissue will be very different. So we will study them separately. So let us start with the plant tissue. Plant tissue can be broadly categorized into two different types of tissue: meristematic and permanent tissue. Meristematic tissue are made up of those kind of cells which have the ability to divide, and permanent tissue is something that has resulted by the maturation of these meristematic cells, and they cannot divide anymore, but they are dedicated to perform a particular function. So meristematic can divide, permanent cannot divide, but it will do a particular function. And according to the location of meristematic tissue in the plant body, as we have just discussed that the growth in plants is localized. So what are these locations? So meristematic tissue, that is dividing tissue, is present in the apical region, in between the mature tissue, that is in the intercalary position, and in the lateral position, and they are known as apical meristem, intercalary meristem, and lateral meristem. Then we go to permanent tissue, and as I just told you, permanent tissue cells are specialized in their structure and function. So if they are specialized, those kind of cells could be similar, or they could be different kinds of cells coming together to take up one particular function. And that's how we divide permanent tissue. If all the cells which are forming the permanent tissue are similar, then it forms simple permanent tissue. And if different kind of cells are grouped together and they are doing one function, it forms complex permanent tissue. And the types of simple permanent tissue that we will study are parenchyma, collenchyma, and sclerenchyma. Here all the cells will be identical in structure. But in complex permanent tissue, the cells will be different types. So first, there are two major types of complex permanent tissue: xylem tissue and phloem tissue. Xylem tissue is mainly involved in water conduction. Phloem tissue is mainly involved in food conduction. And since the definition says that they are complex, so xylem tissue will definitely be made up of different types of cells, and phloem will also be made up of different types of cells which are structurally and functionally diverse. So this is the basic scheme of plant tissue. Now let us talk about meristematic tissue. So first we need to understand that if the cells which are forming this kind of tissue have the dividing ability, then those cells will not be taking any particular kind of structure. They will not be doing any particular kind of function. This is a reserve of cells which can keep dividing. It has retained the capacity to divide so that it can supply the cells for various kinds of permanent tissue formation. And the meristematic cells are characterized by very active metabolism, dense cytoplasm to meet the demands of that active metabolism, thin cellulosic cell walls, because if the cell has to continuously divide, then its wall should not be thick or very rigid, and it has prominent nuclei, because if the cell is continuously dividing, it has to double its genetic content very often, and that's why the nucleus is very active at that moment in those kind of cells, and it has very prominent nucleus, and they also lack vacuoles. So this is very often asked that how will you differentiate cells of simple permanent tissue or cells of meristematic tissue, because after all maturation of these cells will give rise to simple permanent tissue. So you have to start with the metabolic activity rates. the cells which have to divide, they have to maintain themselves in the active stage, and for that they have to synthesize a lot of proteins, a lot of enzymes. So their cytoplasm remains very dense, and they have thin walls for easy division. They have prominent nucleus because they have a lot of genetic content, and they lack vacuole because there is no need to store any kind of food if they are going to divide very quickly. So let us talk about the types of meristematic tissue, and based on their location in the plant body, they can be apical, intercalary or lateral. And let us see in this figure that where generally they can be seen. So apical meristem, as the name suggests, is present at the shoot apex and also at the root apex. So do not forget that apical meristem is also present at the shoot and root apex. Then there is intercalary meristem. So if here is a group of cells which is dividing, it will keep pushing the cells down and they will mature. Similarly, at the root tip, there are a group of cells which are dividing, and they'll keep pushing the cells in this direction so that they mature and start doing their function. But then in between these kind of mature tissue, there are certain tracts of intercalary meristematic cells. If they are meristematic, it shows that they have the division capability, but they are intercalated, that is, they are present in between. In between exactly where? They are present at the base of nodes. Now what are the nodes? So in any plant, the place from where the leaf or branch arises is a node, and between two such nodes, any two nodes, the region between two nodes is known as internode. So intercalary meristem is basically present at the base of the node. At the base of every node, this intercalary meristem will be found. But their occurrence is more prominent in monocot plants. Now, what is their role in monocots? So if we take examples of bamboos, in bamboo, if they are present here, then once they start dividing, they can increase the length of the internode. So in bamboo, they are known to increase the internodal length. And also in grasses, which are also monocots, grasses undergo a lot of grazing. So once their shoots have been taken away by some cow or sheep, then intercalary meristem will help in the formation of new shoots, and that's how the grass keeps growing even after grazing. And yes, one more thing, apical meristems, when they divide, they increase the length of the plant from both the sides. Intercalary meristem in monocots, especially in bamboo, they increase the length of the plant from the internodal region, and they also give rise to shoots which are lost due to grazing, but this phenomena is observed in monocots. And then comes the lateral meristem. Lateral means sides, and lateral meristem is present in the side region. So once this kind of meristem divides, the plant is going to increase in its thickness. So let us see what are they, where are they once again. Apical meristem will be found at the growing tips of stem and roots, and this increases the length of the stem and root. Intercalary meristem will be found near the nodes, and it is active mainly in the monocots. In grasses, it will regenerate the shoots lost due to grazing, and in bamboos, they will cause internodal elongation. And last, the girth or thickness of the stem or root will be increased due to lateral meristem. Lateral meristem's example is cambium. Now, there is one more important point to remember. Lateral meristem is secondary in origin, by which I mean it will not be present since the beginning. Initially, the plant will focus on its primary growth, and therefore we observe apical meristem and intercalary meristem. But when the plant has established itself, it has grown tall enough, and now is the time for secondary growth through which the plant will increase in girth, then only the lateral meristem appears or forms, and then it starts dividing. So the origin of lateral meristem is secondary in origin. This question is often asked, so do not forget. Primary meristems will be apical and intercalary. So this is all about the meristematic tissue, and in the next lecture we will start permanent tissue. So please revise till this far and come prepared in the next lecture. Thank you for your attention.